Assalamualaikum and a very good day. My name is Farzana Nur Hidayah binti Muhammad Hanafiah, and I am excited to share my research on the molecular docking of potential drug candidates against the COVID-19 Omicron variant and its subvariant XBB. COVID-19 has evolved through various strains, with Omicron being one of the most significant due to its high transmission rate. This study aims to explore how we can use molecular docking to find potential drug candidates that could inhibit these virus variants. The main goal in this research is to evaluate molecular interaction in Omicron and Sub-Omicron XBB variant with remdesivir. For the specific objective is to identify the spike protein structure of Omicron and Sub-Omicron variant from the Protein Data Bank (PDB) and ligand from National Center for Biotechnology Information (NCBI). The other specific objective is to evaluate the binding affinity of remdesivir with Omicron and Sub-Omicron using molecular docking technique. Okay, next is before I explain the method in this molecular docking, I will briefly explain regarding molecular docking. Molecular docking is a method that predicts how a small molecule, such as drug, binds to a larger molecule, like a virus protein. In this case, we use this technique to see how well remdesivir could attach to the spike protein of the Omicron and XBB variant. This helps us to understand the interaction between the drug and the virus at a molecular level. In this molecular docking process, they have three main structures. First is extracting and preparing protein structure. Second is molecular docking. And the third one is visualization of protein-ligand interactions. In extracting procedure, first we upload the structure. The spike protein structure for Omicron and Sub-Omicron variants were extracted from PDB, protein database, and the ligand was extracted from NCBI. And then both are uploaded to the Galaxy server. In pre-processing sample, for protein, we first utilize the grab tools to extract protein atom. And then selected line from 6VVX.pdb, which is an Omicron protein that extracted from the database, and 8ios.pdb is a sub-Omicron, which is XBB, and excluding the HETAM. HETAM is basically a non-protein atom. And then for the ligand, first is convert the ligand PDF file to MDL mode format using the compound conversion tool and set pH to 7.4. Next step is preparing for docking. The protein use the prepare receptor tool to convert the preprocessed file from SDF to PDBQT. And then for the ligand, configure ligand docking parameter using RDKit, including setting, buffers, and adding hydrogen for pH 7.4. Next is molecular docking. In each PDBQT protein structure was docked with the ligand using specific parameters, including receptor file, ligand SDF, pH 7.4, and configuration file. And then the last step is visualization of protein-ligand interaction. First step is load structure into PyMol. Use the protein structure that already been in the PDBQT file and dock ligand structure into the PyMol. And then use the align command to align docked ligand with the Omicron protein structure and another docked ligand with Sub-Omicron protein structure, both also in PDBQT file. And then display the align structure and visualize interaction with the show stick and show surface commands. Now let's delve into the result of this study. Table 1 shows the details of spike protein that being used in this study. Both of the samples were extracted from Homo sapien, and both of the spike protein have three identical functional proteins, which are chain A, B, and C. However, spike protein of Omicron have longer amino acid chain than XBB variant, which are 1,281 amino acid for Omicron and 1,045 amino acid for XBB. Both of spike protein are being constructed in 3D structure by using electron microscopy. Electron microscopy is a high-resolution imaging method which allows it accessible to view the atomic structures of the protein. Table 2 shows the docking result between spike protein of Omicron and XBB variant with remdesivir. From the table 2 also, Omicron spike protein, which is 6VXX, has been docked score of negative 182.351 kilocalories per mole, which is more negative compared to XBB 1.5, which is 8 IOS, with negative 153.191 kilocalories per mole. Moreover, Omicron's H-bond energy is negative 1.19817, whereas XBB has a stronger H-bond interaction with an energy of negative 4.91563. Based on previous study, higher number of H-bond correlated with a lower docking score. It indicates stronger and stable molecular interaction. Table 2 also provides the amino acid of both spike protein that interact with ligand. For Omicron spike protein, the ligand interact with amino acid that in position 61, 27, 63, 28, 65, and 62. Meanwhile, for XBB variant, the amino acid that interact with ligand are in position 20, 21, 19, 158, 140, 244, and 258. Both interaction is happen in chain A of spike protein. This interaction can be seen in figure 1 and figure 2. Blue color of molecules in figure 1 are spike protein of Omicron, and green color of molecules in figure 2 are spike protein from XBB variant. The red molecules in both figures are showing the molecules of remdesivir. The molecule that in grey color for figure 1 and light blue for figure 2 are the amino acid of both spike protein that interact with the ligand. Previous study mentioned that chain A from both variants of spike protein specifically interact with the ACE receptor. So, by the finding of this research, remdesivir can interrupt the invading process of this virus from entering the host cell. In conclusion, based on the molecular docking result, both spike protein variants demonstrated significant binding affinity with the remdesivir. The result indicate that remdesivir effectively binds to both Omicron and XBB spike proteins, suggesting its potential as a therapeutic candidate. Overall, the study highlights remdesivir's capability to bind effectively, providing valuable insight into how specific mutation might influence drug binding. I would like to end my presentation with an acknowledgement. First, I would like to express my deepest gratitude to the Faculty of Health Sciences, University Selangor, and also staff Immunoserology Unit, Hospital Kuala Lumpur for the support and their guidance. Thank you.